The tissue obtained from a plant to be cultured is called an explant. Explants can be taken from many different parts of a plant, including portions of shoots, leaves, stems, flowers, roots, single undifferentiated cells, and from many types of mature cells, provided they still contain living cytoplasm and nuclei and are able to differentiate and resume cell division. This has given rise to the concept of totipotency of plant cells. However, this is not true for all cells or for all plants. In many species, explants of various organs vary in their rates of growth and regeneration, while some do not grow at all. The choice of explant material also determines if the plantlets developed via tissue culture are haploid or diploid. Also, the risk of microbial contamination is increased with inappropriate explants. The first method, involving the merisms and induction of multiple shoots, is the preferred method for the micropropagation industry, since the risks of semiclonal variation are minimal when compared to the other two methods. Somatic embryogenesis is a method that has the potential to be several times higher in multiplication rates and is amenable to handling in liquid culture systems like bioreactors. Some explants, like the root tip, are hard to isolate and are contaminated with soil microflora that becomes problematic during the tissue culture process. Certain soil microflora can form tight associations with the root systems, or even grow within the root. Soil particles bound to roots are difficult to remove without injury to the roots, that then allows a microbial attack. These associated microflora will generally overgrow the tissue culture medium before there is significant growth of plant tissue. Some cultured tissues are slow in their growth. For them, there would be two options: optimizing the culture medium, culturing highly responsive tissues or varieties. Necrosis can spoil cultured tissues. Generally, plant varieties differ in susceptibility to tissue culture necrosis. Thus, by culturing highly responsive varieties, it can be managed. Aerial explants are also rich in undesirable microflora. However, they are more easily removed from the explant by gentle rinsing, and the remainder usually can be killed by surface sterilization. Most of the surface microflora do not form tight associations with the plant tissue. Such associations can usually be found by visual inspection as a mosaic, decolorization, or localized necrosis on the surface of the explant. An alternative for obtaining uncontaminated explants is to take explants from seedlings which are aseptically grown from surface sterilized seeds. The hard surface of the seed is less permeable to the penetration of harsh surface sterilizing agents, such as hypochlorite, so the acceptable conditions of sterilization used for seeds can be much more stringent than for vegetative tissues. Tissue-cultured plants are clones. If the original mother plant used to produce the first explants is susceptible to a pathogen or environmental condition, the entire crop would be susceptible to the same problem. Conversely, any positive traits would remain within the line also.